Hello. Hey. What happened to your CCA? How I thought your CCA like will make you come back late. That's hell the sus, but okay, sure. That's a bit sus, but okay. Then your bio paper, where do we lose marks? Where the marks fly away to? Good question, yeah, lah. Good question. Good question, just so you don't know. Yeah lah, but what topic is it? Is it like cells? Is it like osmosis, diffusion? Huh? What's a different topic? You only tested on two topics. What's the three topics? You only have tested two topics, right? Chemistry. You're telling me chemistry marks are back? Okay, but still go back to the same thing. Then what do we lose marks on bio? Why you very good at talking nothing, you know, and you're good at making nothing burgers, bro. Huh? So you have not. No, most likely you saw the paper, but you didn't care about remembering where you lose marks. Yeah, you did the corrections, then you should know where you lose marks, what? Sure, but still go back to the same thing. Where do you lose marks? You just yep that you say you did your correction. Now you're talking nonsense, bruh. You're full of shit. Then what part of the cell structure, man? I mean, do you have the paper with you? You most likely will say no, right? Oh my god, you're full of shit. I don't think. I mean, I'm sure. I mean, do what you need to. Yeah, sure. Because 20 out of 30 is like, I mean, in my books, that's borderline pass. 15 is a pass. 20 is like very close to like pass fail line. Not a good look though, especially you got so little to study. Only two chapters. So I want. Huh? Uh. So you are telling me 20 and 26 is not that far away? Are you seriously? Are you saying that? 26 is a lot more than 20 when it comes to the total marks 30. Whatever you say doesn't make any sense, bro. I don't know what you're talking about. Because temperature and is that bio or is it chemistry? Speed up what experiment? Is it chemistry or is it bio? Speed up reaction? What reaction? What process? I tell you what, you become politician or become scammer lah. You damn good at making nothing burgers, bro. Be a crypto bro lah, yeah. Talk many, might as well don't talk. You know, mean jack shit, bro. We're talking. Uh huh. Uh huh. What? One side got something, then something, then something, 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 something. That's what you said. You said nothing, bro. No details. You're just filling it with a nothing burger. Let me repeat, go back to the same thing. Is this chemistry or is this bio? Yeah, that sounds like chemistry to me. Movement of substance in bio, that diffusion. So? Whatever you said sounds like a chemistry thing, not a bio thing. I seen though it. No picture, no proof, bro. That's all I can say, man. So what you're trying to tell me is, you got a paper bag, you did correction, and you remember nothing. Okay? Huh? Yeah, that's what you're saying. You can't tell me any details, bro. You do not care about the correction, therefore you remember nothing. You got so what? You got it back yesterday, so by today, because it's like 24 hours, therefore everything evaporate from my head. Nothing but just TikTok dancers are legit. Oh my god, really? Come on, man, that's full of shit. Oh my god. Uh-huh. I don't know. I think you're cooking random words and then you just randomly say something for me to like okay and just not talk about it. That's what it is, bro. But cannot be the whole cell got like multiple nucleus, right? Because if mitochondria, I'm pretty sure there will be multiple mitochondria, right? Okay. Then what's the latest update when it comes to your chem and your bio stuff? This is chapter 1, chapter 2. What's chapter 3? Digestion. Is it? What's chapter 4? Okay. Today is on the 27th. Okay. Hello, you there? Yeah, so what is chapter 4? Google. Oh my god. If you follow okay. Because if you follow the chapter by chapter scenario, we are looking at nutrition, which is digestion. That is if they teach enzymes. So how? You found the fourth chapter? Have you found the fourth chapter? Hello? Mosh mosh. Okay, then restart your com. Hey, you're back. Okay, so what's the fourth chapter? Of course. Okay. Then chemistry? Sorry, what? You there? Hello. Mosh mosh. Mosh mosh. Hello. You there? But I can't hear you. Something's wrong. I don't know, man. I mean, at least Discord is not lighting up. Yes, now I can hear you. Okay. So yes, what about chemistry? Okay. Chemistry? Okay. Then chemistry? What? That's all. Chemistry, did it stop at atomic structure or what? So last chapter is atomic structure. So no ionic covalent stuff. Yeah, no ionic covalent stuff. Okay, then that's fine. Atomic structure. Okay, in this case, I think I want to do the bio stuff. We are behind schedule from your school stuff. Okay. Do you... Nah, I think it should be fine. Wait a minute. No, the chapter is small. Food test only has like 4 things. To test for starch, to test for sugar, protein, fats. No more already. 4 things to talk about. Enzymes, talk about lock and key, which is repeat of what Sec 2 is. Then after that, temperature, pH, denature. Nothing much, right? You're just learning what you already learned, right? What do you mean by that? Yeah. Yeah? So you say it's different, right? So what do you mean by that? Okay. Yes, substrate. Complementary. The shape is complementary. Then? What do you not understand? What do you mean? You're saying something very general, man. And every time you say something like this, come on lah. You're really smart and coming out with bullshit. I know that you know that, huh? So don't come and bullshit me. I'm not that dumb. So yes. So yes, what do you not know about it? Then what? Then what's and that's what? You're just stalling, man. Oh my god, man. Okay. Then what's enzyme? What is enzyme? Let's start with the absolute basic, the beginning of the chapter. What's enzyme? Okay, enzyme is used to speed up chemical reactions. Other than that, and so it's used. So number 1, it is speed up chemical reaction. Number 2, it is not chemically change. So it have to fulfill 2 conditions. It has to speed up chemical reaction, and it has to not change chemically. So you will not become nothing. So you can reuse enzyme. Then what is lock and key principle? What is lock and key principle? Okay, so lock and key principle. Maybe I just find a picture. Oh my god, this looks kind of cute. Okay. Yes, this is a lock and key principle. In other words, why is the ad so big? Okay, so what happens is that like this is the enzyme. Enzyme, of course, it has an active site. Active site is basically the location where the substrate will stick into it. So substrate will go into the enzyme and fit at the active site. When they combine, the whole thing is called enzyme substrate complex. These are all important words you have to write, or else there's no marks given in your essay question. So when they bond together, it becomes enzyme substrate complex, and then it forms a product. Because just now from this, now it splits in half, so the product is released from the enzyme. That's all enzyme is. So usually enzymes in your syllabus, and the enzymes in your syllabus usually focus on digestion, which you learn before for like, I think Sec 1 or Sec 2, one of them. I'm pretty sure you learn about that, but I'm pretty sure everything also forgotten already. So yeah, so that's enzyme in general. And enzyme is going to be affected by, so lock and key principle means that the shape of the substrate will fit the enzyme specifically. So that means that one type of enzyme is only going to fit one type of substrate, just like a lock and a key. Does that make sense? The whole thing is the enzyme. The whole thing is an enzyme, and in the enzyme, the whole that fits the substrate is called active site. Yeah. So yeah, so that's that. So so then enzymes can be affected by mainly two things. One can be affected by temperature, and two, it can be affected by pH. So when it is affected by temperature or pH, the keyword is called denature. So question, what is active site again? So active site is the location on the enzyme. So let's say now this is the enzyme. So if this is the enzyme, this is the active site. So a substrate can fit into it. So this is like the substrate. This is the enzyme, and whatever that fits into it is the active site. So when temperature or pH cause the active site to change shape, so the active site shape can be changed by high temperature or very different pH, we call denature. The word denature, denatured, means the shape of the active site is changed, and substrate is unable to fit properly. So when properly, so what happens is that if the active site shape is changed, then the substrate cannot fit, unable to fit. Yeah. So yeah, so that's that. Okay, we will try questions. So at this rate, will your chemistry marks be better than your bio? Light can pass through. Why light can pass through? That is chemistry. Ghost shit. What's a ghost shit? Oh, okay. Gold. That is a gold Rutherford gold foil experiment. So, so what does it talk about? Oh my god, what else can you remember, bro? I mean, okay, that's fair enough. Okay, sounds good. Okay, let's do this, but you need to inclusive of both uh, digestion, enzymes, and food test stuff. So yes. Nice, okay. This is a common question, so should be fine. Mm. Okay. Okay. No, root has only takes in water and mineral salts. Hmm? What a comparison. One takes in oxygen for both. No, nah, I think the answer got problem, right? Wait a minute, let me double confirm. Root hair cell. Root hair cell don't take in oxygen. Cytoplasm, yes, yes. So large surface area to volume ratio, yes, yes. Nucleus present, no, yes. Answer should be should be D. I don't think it root hair cell takes in oxygen. Should be a yes, no. Okay, how to reset this? Move on to the next one. This one is question 3, 4. Okay, question 4. Refresh on your side, it should be correct already, so yeah. Mm-hmm. So at what position will there be no net movement? It says here sucrose, what? It says here sucrose solution, what? Yes, applied to this question. Where is there no net movement to this graph? Huh? What are you talking about? Well, then you don't know how to read this graph at all, bro. What is this graph about? So what do you see? Yeah, you see nothing. What do you see? Tell me. I also can see that. My eyes also can see. So now, my question to you, I restated the question for the 4th time. Where is there no net movement on the graph? At what point? Then no, I'm gonna ask you why. You don't cook up random shit to me. Where's the straight line? Where's the straight line? 0.8. 0.8 is here. 0.8 here. Okay, why? You're not explaining shit, bro. I'm asking you why. Give me the reason how this line is linking to no net movement of water. You're not giving me any explanation. Anything with eyes, with mouth. If not, anyone who not blind can tell you lah. Anything else? Oh, this line here, 0.8, 1. Come on, bro. You're in pure science. Plus more lines means that basically the cell. Let me just draw this lah. So when you have, okay, then what's plus more lines? What shrink? What shrink away from the cell wall? Perfect. That's a good explanation. Oh, that I don't know what you're talking about. What does that last sentence about? Go out what? Go out from where to where? As in, you're okay. In this graph, what is the graph about? Read, bro. Read. Is reading now for dummies? Read. Equal volume of 5 different concentrations of sucrose in 5 different containers. Everything looks similar. Identical piece of tissue placed in every one of them. Plant tissue will remove for 45 hours and change in volume of solution. So what does this mean? Okay. So why would the sucrose solution volume change? No. Let me repeat what I'm asking. Why would the volume of the solution change? Yeah. Why would it somehow change? In this case, no negative number, so the volume keeps increasing. Somehow, yeah, why would the volume just increase for no reason? Water potential. You don't use water concentration. So, and then higher water potential. So, then. Okay. So then my next question is if I want to find a position where there is no net movement of water molecule, what will be the volume of the solution? Let me correct that. What will be the change in volume of the solution if there's no net movement of water molecule out of the plant tissue? So therefore, what point would that be? It will be here. This will be the concentration because it's the volume. Let me or maybe let me just draw this. That means if there's no net movement, the change of volume will be 0. Make sense? Am I making sense? If there is no net movement of water from the plant to the solution, my change of volume will be 0. Make sense? Or doesn't make sense? So anything below 0 is negative numbers, huh? Yeah, sure. Because the opposite can happen, right? Water can actually enter the cell, right? Then your volume decrease, right? So let me repeat my question for the third time. Yeah, I'm very good at not answering my question. I'm asking the. Okay, what am I asking? What did I ask? Yeah, talk. What do I ask? You say you don't understand, right? I say you're not listening. What did I ask for the third time? Yeah, what did I ask? Okay. So what part do you not understand about the question? Or rather, when I drew on that red mark there, what does it mean? Because I was mid explaining that thing. So what am I explaining? Yeah, what happens when the change of volume is 0? What does it mean? No water increase. Okay, why is the water not increasing? Or decreasing for the matter? Equal between? Yeah, on the right track, but equal between what and what? Perfect, yes. So therefore how do we find the concentration of the plant tissue? Any idea? This is new stuff. How do we find the concentration of the plant tissue? So I don't know. I didn't. Yeah, I think so. I think it's 0.8 because I think halfway is like 0.1, right? So this should be like 0.08, right? Make sense? Yes? No? Yeah. And I'm literally trying to explain things that will come up for your exams, because I heavily suspect something like that did come out, and then you make mistake, because questions of graph like that is very common. So let me confirm one last thing that I explained just now. Why, as what you said, right? Why is there negative numbers? Because anything and this black square here. Correct. So therefore, the water will actually enters the cell and cause a decrease in it. Make sense? Okay, perfect. So, can you explain why is the answer B? Can, then I reset it. Why is it B? Would you want to explain why it's B, or you want me to just skip it and move on to the next question? I'm okay either way. So how? I. Hello, you there? Can you hear me? So I can't hear you, so it's just saying. Hello, you there? Hello. Oh, okay. I can't hear you. I mean, assuming that you, I don't know whether you can hear me. Hello. Hey, okay. Okay, so let's move on. Question 5 then. The what? Yeah. You didn't ask me a question I cannot explain, my dear. Okay, what's plasmolyze? Yeah, what is plasmolyze? Yeah, what is plasmolyze? Hello? You there? Ynessa? Hello? Yes. Okay. Okay, so my question to ask you is just now, what's plasmolyze? Then it says that 0.8 and 1, so 0.8 is here, 1 is here. When the concentration is very high, what does it do? When the concentration of the sugar solution is very high, how does it affect the plant cell? Then clearly now this is one straight line. Why doesn't the water loss change? Why is it just one straight line? Yes, all the water has leaked out, right? There's no more water leak out anymore, so how there cannot be any difference in change of volume anymore. Okay? Okay, question 5. I don't know. You are damn good at just spinning nothing and nothing burgers and nothing burgers and nothing burgers. Then you do this. I just now say, then there's like 5-10 minutes ago. I don't know what you're talking about 5-10 minutes ago, man. You most likely tell me about water move in, water move out, but you didn't apply to the question. So yeah, so that's what it is. Question 5. You know about question 5? What it's about? Nice. Okay. Then this is, I think this is osmosis, but because it's got partially permeable membrane. Okay. Then glycogen and polypeptide, that's the food stuff. So, okay, that's nice. Then this is on anything that's digested can pass through. Anything that's not digested cannot pass through the partially permeable membrane. So when lipase digests the fat molecules, whatever is digested can pass through. And just to confirm that, fats get digested by lipase to form fatty acids and glycerol. So that so with fatty acids, then you'll cause the pH to drop. You become acidic. So pH will drop because if there's fatty acids. So from 7 it drops to 6.1. So the only thing that pass through the membrane is going to be anything that's digested. So only fatty acids and glycerol can pass through the membrane. Nothing else can. Fatty acids, true, because fatty acids diffuse through, that's why the pH dropped from 7 to 6.1. Yeah, so because it's digested, therefore it only that is able to move out. Okay, that's good. Okay, then okay, this one, right? Just to let you know, Q has different shapes. Different shapes means that it's a protein. The one that looks like a jellyfish, that's a fats. The one that is just repeat same shape again and again and again, that's carbohydrates. Q is protein. Yep, Q is protein. Yep. Enzyme is made of protein, but not all proteins are enzymes. Then why is D correct? So, okay, I'll reset this. This one, okay. Then same thing. This is the different food substance stuff. Okay, give it a try. please ask me about the functions and stuff. So yeah. Carbon, hydrogen, and oxygen. Fat is used to store energy in the body. Mm-hmm. Nice. Okay. Okay. Fat should produce the most energy, then followed by carbohydrates, then followed by proteins. True, which is fats in this case. Then, okay, let's do one more. Question 12. Okay. Nice. Okay. Okay, then we are done. There's no deadlines or anything coming, I assume. Homework, deadlines, projects. Don't have, Just checking. Okay, then I guess see you in a week's time then. Bye bye. Bye bye.